functional spectroscopy and brain stimulation. So yeah, just keep it interactive. Maybe if there's questions along the way, we can take one or two, but then keep the rest to the end. Ian is from the Melbourne Neuropsychiatry Centre, which is misspelled inside recently finished his PhD and did really dug into the nitty-gritty of functional imaging, and he really knows his stuff, so he's going to tell us a bit about that.
Thank you, Matt. So, I think we've kept everybody, which is great, actually. Everybody's doing very well. So, what I would like to do this morning is a very similar grain to what we've seen already, is talk about fMRI, talk about what it is, what we're actually measuring, what is this thing called fMRI. I'm going to sort of leverage off of what Paul was talking about this morning with MRI, and add that little F on there. Then we're going to talk about blood maps. There's what we lovingly call blood maps in the field, these brain activation maps. How do we get there? What do they actually mean? And in terms of pathology, I've got some examples of some blood maps that show pathology in some various psychiatric illnesses and what that might mean for our interpretation. And then we're going to touch on brain connectivity, which is all the rage at the moment. And this is functional connectivity, as opposed to what Monash was talking about in terms of anatomical connectivity. Anatomical connectivity being the white matter connections, the actual axons in the brain. Functional connectivity, we'll get there, but it's more about how brains interact in a functional sense. So, getting an fMRI, we know from what Paul was talking about that MRI indexes the magnetic properties of the brain, particularly something we call susceptibility. So susceptibility in terms of structural or static MRI is determined mostly by the local water concentration. So more or less water gives us that gradient that we see in the structural MRI. In terms of the dynamics of the susceptibility of the brain, that's primarily determined by changes in blood flow, which is something that we can tap into. So how does that actually work? Well, when a neuron starts to fire, or I should say more accurately, when a neuron changes its firing rate, it increases metabolic demand. The brain doesn't actually store oxygen or glucose or other requirements of metabolism, and it doesn't have a mechanism to store the waste products either. So we have this amazing neurovascular coupling in the brain, where as soon as we get a change in metabolic demand, we get a change in blood flow locally in those capillary beds. Along with that comes an increased blood flow and increased oxygenation. In particular, we get an increase in oxygenated hemoglobin. So hemoglobin being the protein that carries oxygen in the blood, in the red blood cells. And so I have no formulas in this talk, but I did get a bit of biochemistry in there, sort of priming for Chow, who's coming up next. So don't worry about the details here, but this is a heme group. And what I want you to notice is that in deoxyhemoglobin, when it's not bound to oxygen, it's ionic. It's got this 2+. When it's bound to oxygen, those free protons are bound up by the oxygen itself, and it's neutral. And therefore, the difference between, therefore deoxyhemoglobin and oxyhemoglobin affect the magnetic properties of the brain in different ways. It's very, very small, it's very slight, but it's something that we can measure. And so as oxygen comes rushing into this local capillary bed, we get an increase in oxyhemoglobin. The change in the ratio of oxygenated hemoglobin to deoxygenated hemoglobin gives us an increase in what we call the blood oxygen level signal. I'm just going to call it the fMRI signal. And that's what we actually measure underlying to get a measure of function. And this is directly proportional. This signal is directly proportional to the change in neuronal signal underneath. As opposed to an anatomical study where we get a snapshot of the brain, fMRI relies on signal variability measured over time. So we have this repeated measures designed for two reasons. One, the brain is never off. And so if we want to measure function, we need to compare it to something. So we need two conditions. It's always repeated measures. We have what we would call a baseline, we have an active condition. The other thing is the brain is incredibly noisy. So this is actually, if we take one voxel in the brain, so we divide up the brain into tens to hundreds of thousands of voxels, usually on the order of about 3 millimeter cubed, 3 by 3 by 3. And we measure each of these voxels independently over hundreds and hundreds of time points. So each one of these blue dots is actually one time point. that we've measured. Each one occurs about 2 seconds, 2.5 seconds in this case. The black line, I've just temporarily smoothed it, so it's a moving average of about 5 of those points. So you can sort of work through the noise and see that there is a pattern in that data, and I'll talk about that in a second. But all of this noise means that we really need to get a lot of measurements to actually get something meaningful. We can actually tap into that noise in terms of functional connectivity as well, which we'll touch on here in a little while. So, in terms of brain activation, how can we leverage this to come up with something meaningful, or to investigate the brain meaningfully? Well, as I mentioned, we need at least two conditions. I want to say the sky's the limit. You can kind of do a lot of things. There's a lot of scope. Anything from basic perceptual, motor, emotional, cognitive. This is a cognitive task called the flanker, where you're responding to the middle arrow, whether it's left or right. Language is my gibberish manipulation. So you have task A and you have task B. And you're needing to introduce the variability in terms of your psychological manipulation to induce variability in task brain regions. So down here we can see that we have task A going for, call it 15, 20, 30 seconds, and then switching to task B. Task A, task B, task B, task A, task B, back and forth. And we're looking for reliable prediction of the fMRI signal in those voxels based on the psychological manipulation that we introduce. And in this case, we can see very clearly this. I actually selected this particular voxel because it was tasked meaningfully, that we can see that there is a difference in the signal strength from task A to task B. We then take the subtraction of those two. We find the average of what's happening at task A and the average of what's happening at task B, and we can say that is the strength, or it provides you with a proxy measure of the strength of the neuronal activity in task-relevant brain regions. We can do that across the entire brain in every single voxel independently, and we come up with effect size maps. So we love to use colorful maps in functional imaging. It gives us high impact computation. But it's also very useful, because otherwise each of these colors just represents a number. In this case, it's an effect size. Blue is actually negative, so it's the flip, where B is greater than A, whereas all the red and the orange is A greater than B. And we just use these colors as a nice representation of a statistical map. So we start with all voxels in the brain, or we can have a region of interest, if you will. And then we threshold it, so this is estimation, and then we undertake statistical inference to get our P less than 0.05. And we find these regions that we would call statistically significantly activated in a particular task, or within that subtraction. And these are our blog maps. These are the nice pictures that the media loves. These are the nice pictures that we publish. But we could very easily just change out each of these individual boxes for a number, or a bar graph comparing our two conditions. That's what we're looking at. And there should always be sort of a scale along with it, showing you if it's a T statistic or an F statistic, or whatever it might be. So that's what we're talking about in terms of activations and how we get there. In terms of pathology, we can then compare our activation maps for a control group versus any pathological group. We can do it on the individual level as well, but generally in psychiatry we're interested in looking at population level differences on average. We can get either both qualitative and quantitative change in signal strength, in neuronal activity. Generally, quantitative reductions are thought to represent a neuronal dysfunction in a particular region. On the left, we see schizophrenia. This is actually a meta-analysis of 41 executive functioning tasks. And we can see that we have a very similar pattern of activation in controls as we do in patients. But that in particular, we see a reduction in activity fairly consistently in dorsolateral prefrontal cortex, in this case weighted to the left, being an executive functioning tasks. We also see reductions in medial structure, but we also see increases in activity, represented here in blue, that suggests that there might be either a compensatory mechanism going on in these regions, or that these regions are functioning, they're doing what they need to be doing, but they need to be activated more in order to actually get there. Oh, the other thing I wanted to mention before I move on to the other, well, this is a study from Monash and colleagues looking at OCD, and in this case it's very similar. We have controls on the top, OCD patients here in the middle, and we have a very similar, just if you look at this image, very similar pattern, and it's tough to see, but you do see significantly greater activity in medial structures in particular, and a bit down here in the insula as well. But in psychiatry, generally, this is what we see. We see a similar pattern of activation, where we see quantitative difference between the two. But we can also have a look at qualitative differences. This is the best example I could find of some data we're working on at the moment, where this fellow had a medial prefrontal resection because of intractable seizures. But we know that this part of the brain is really is necessary for certain cognitive tasks that we are interested in. So we popped this guy into the scanner, and we were amazed that he could do the task. He was on par with a group of controls as 20 controls, but what we saw was the pattern of activations were different. In that now no longer these blob maps no longer look the same, in posterior structures certainly, but if you see here where you have a lot of lateral prefrontal activity, we don't really see that lateral prefrontal activity in controls. Whereas, well, obviously, I mean, you see the medial activity here, doesn't even have brain tissue there in order to do it. So he has this compensatory shift or a qualitative shift in activity structure, which sort of allows us to perhaps understand some of the ways that he might be compensating for this lost tissue or the lost abilities that would be inherent to him. I believe that it would also be much more relevant to neurological disorders where we have a particular lesion in the brain, as opposed to these diffuse psychiatric type of illnesses. So moving from activation onto connectivity, we now are no longer interested in each voxel in the brain, but how voxels interact with each other. And so in this case, we can have a look at region X, which could be a single voxel, or generally we would use a larger sort of average of a bunch of voxels. In region Y, we would extract the signal from each of those and correlate them. This correlation, for example, I just made this up to be honest. This correlation, 0.36, would suggest the magnitude of a functional connectivity, or how much shared variance there is between these two brain regions. We can then create these nice matrices. We saw some of Monash's talk as well. To be honest, I mean, we're all friends here. I actually only recently figured out what the heck this means. So you might see this in terms of functional connectivity, even anatomical connectivity. And so I'll explain it briefly. Just every region of the brain is represented in a column, so we have A, B, C, D, all the way down, as many as you have, and by a row. And so the connection strength between, say, region, in this case, region X, which would be represented here, and region Y, is a 0.36. And again, we use colors because we're functional neuroimagers, and it makes us happy. So we have this nice sort of color map that gives us a connectivity structure of the whole brain. So we can stop here if we're only interested in these two regions, but we can do this across every region of the brain, or every pair of voxels in the brain, or every pair of regions.
Sorry, can I ask a really quick question?
Yeah.
Then the high correlations are clustered together. That's based on a spatial...
Yeah, it's a topological structure. So in this case, this is not ordered, it's not ordered arbitrarily, sort of A to Z. It's actually ordered based on lumping regions that form networks together. And so we'll see that down here a little bit more, and then this is the same now matrix. And I wanted to make the point that we have a distinction between resting state connectivity, where somebody's just lying in the scanner doing nothing, we generally acquire for 6 to 10 minutes, and we're using those spontaneous fluctuations of the brain. So we're not introducing a task in this case, we're just saying we know that the brain is very active intrinsically. And so we're looking at correlations in those intrinsic fluctuations, which is nice because then it's no longer task specific. It's sort of a general property, intrinsic property of the brain. And over the past 5 years particularly, there's been an explosion of research in resting state connectivity in pathology and psychiatry and neurology, because in our quest for biomarkers, this is nice that we don't need to introduce a task that patients may or may not be able to do very well. So this is just showing that we get a clustering of regions into what we would call resting state networks of the brain, which is something that we see a lot in the literature now, or intrinsic networks of the brain. And I won't go into the details, but these are sort of the classic intrinsic networks. When we're talking about task-based connectivity, there's a number of ways to do it, but generally what we're interested in is how regions are interacting during task A and how that interaction changes during task B. So if we can change in the strength of the interaction, it suggests that these regions are task relevant. And so in this case, again, we have region X, region Y. This is a lateral prefrontal region. This is a occipital parietal region. I'll show you this study actually in a second in more detail. And during task A, we don't have a significant correlation that we get a significant change to a correlation during task B, suggesting that this interaction becomes relevant to whatever the difference is between A and B. Yeah.
So even at the resting state, there needs to be a change for it to be indexed to other regions.
So we still measure over time. We still use repeated measure design. We collect hundreds of measurements across our 6 or 10 minutes, and then you're using spontaneous fluctuation, spontaneous change in the brain signal, and correlating that instead of a particular task signal. So a couple of examples of using connectivity. Again, we can have quantitative or qualitative changes. Quantitative changes generally thought to index abnormalities in synaptic efficacy or heavy in learning, so neurons that fire together, wire together, or axonal conduction, which goes back to anatomical connectivity that Monash was talking about. And this, again, we often see quantitative differences in psychiatric illness. This is a nice study done by Chris Davey, who is a psychiatrist here, in a depression cohort. And in particular, he was interested in how regions of the cingulate cortex, particularly this anterior limbic region of the cingulate, interacted with the rest of the brain. So he wasn't doing pairwise between every region of the brain to every other region of the brain. He had an a priori region of interest because of known pathology in this region in depression. And he found that the maps are similar, so I know you can't really see this. Green is control connectivity, red is depressed, and yellow is the overlap. So we see a lot of yellow, which suggests the connectivity profiles are qualitatively quite similar. I didn't show the quantitative difference, but particularly from this seed in the pregenual ACC, he found a significantly increased connectivity to orbital frontal regions, was the primary finding of this study. Ben Harrison in an OCD cohort did something very similar, but his a priori hypothesis had to do with the striatum. And so he, what we call, seeded the striatum and looked at how that interacted with the rest of the brain. He actually found something quite similar, particularly for the ventral caudate, that there was, and again here, yellow is the overlap, red is the OCD, and green is the controls, that there was a significantly increased connectivity between the striatum and the orbital frontal cortex in OCD. So moving beyond the functional activations, or how particular regions of the brain are activated by attacks, to now looking at the network structure of the brain. Because of course, regions of the brain unto themselves don't provide meaningful behavior. It's that connectivity that's really important. And particularly in disorders in psychiatry, where we're seeing these qualitative similarities in activation profiles, and we don't have a particular lesion to work with, perhaps connectivity is a nice way to get at the more diffuse pathology that may be involved. A couple more quick examples. In schizophrenia, we often don't have the same sort of a priori hypothesis. It doesn't seem to be within reason. I mean, DLPFC, or dorsolateral prefrontal cortex, seems to be one of these regions that comes up consistently in schizophrenia. But in general, we're still working at the whole brain level. So this is a study done by Alex, and looking at both, in this case he took a little bit of a different approach, but looking at task-independent connectivity, and found that in schizophrenia, it's tough to tell here, but it's kind of a generalized decrease across the whole brain in connectivity, particularly within frontal temporal regions. But when you sort of remove that generalized deficit, when he did a cognitive task, he found that there was further disruption, particular to task-related connectivity in additional regions. And this maps quite nicely onto the fact that in schizophrenia we see this sort of general cognitive decline, and then over top of that general cognitive decline there's particular executive functioning deficits and these kind of things. And this sort of gets at that general decline as well as the specific task-related decline. It's a very nice word. And this is that task-related connectivity going from this lateral prefrontal cortex to the occipital-parietal cortex that I had shown a couple slides previous. And this was actually a study that we did in cannabis users. And we found that this connection, which is thought to sort of drive attentional processing, these sort of things, was modulated significantly in cannabis users during an executive task, but not in controls, indicating a task-specific hyperconnectivity of this connection. So that's actually all that I have. I'm happy to have a bit more discussion.
Great, good timing.
Thank you.
All right, the floor is open.
Yes.
So, when you look at fMRI, do you also, in the scanner, take the MRI at the same time, and then you somehow put the signal onto the structural image? Is that right?
So we actually do it at separate times. So we get our what we call a T1 weighted image first, which is our structural image, and then we would do the functional afterwards. It's kind of getting at, it uses slightly different properties of the MRI signal, and sort of leveraging this idea of we're interested in water in the structural versus we're interested in this change in the hemodynamic response in the bowl. And so so the structural images provide a nice template that we can then lay our functional images over top of. The other thing that's important is, of course, people have different sized brains. I mean, the brains are in different space, we like to call it, in the field, and so we need to put all of the brains, we need to map them all into the same space. And we can do that with the fMRI images, but they're of a very low resolution. We don't get a very good sort of overlap, if you will. So we often use these high resolution, high spatial resolution structural images in order to register all the brains together, and then we can overlay the function on top of that, or apply the same transformations to the function, so that we can then do group level studies.
And doesn't it give it a task, give someone a task? What's the time frame that the task has to be doing?
Yeah, that's a good question when it comes to task design. So there's a lot of properties of the fMRI signal that introduce noise. There's physiological noise, there's scanner drift, we call it. And so we can't do sort of long extended periods of one task and then long extended periods of another, because that gets lost in our noise. Generally, we try to kind of get 20 to 30 seconds of a block, if you will, and switching between blocks. You can go up to about a minute. Beyond a minute, you're probably starting to run into problems. And that's for a block design, which is sort of the most powerful design. If you're looking at doing studies, I would suggest start with the block design, start simple. I did a bunch of really complex stuff in my PhD, and start simple, please. There's nothing wrong with the block design. It gives you that nice power. You can get an effect and then start to parse it out. The other is called an event-related design, where you're, instead of having a block of similar stimuli, you're presenting one at a time, and you're intermixing them. And so you lose statistical power, but you can then issue problems like predictability of what's coming next, or some things like auditory oddball that Nigel was talking about, boop, 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 beep, boop, boop, boop. You can't do that in a block design, so you need that event-related design. So it really depends on your questions, but yeah, block design is probably the way to go if you're first getting into an effect, and try to vary it in on the order of 20 to 30 seconds.
We have tasks at the moment that do go up to half an hour. Sometimes when you're doing learning paradigms, it takes a while to learn things, and you just got to be very careful the way you design them to not, like what Ian was saying, kind of just make sure you're not acquiring a bunch of noise. It has to be designed in a way that you can actually do those long half an hour type studies.
That's a good point too. So I was talking about blocks sort of switching for 20, 30 seconds, but you need to scan generally on the order of 10 minutes in order to get a reasonable signal to noise. So attendance is kind of the minimum. You can split that up. You can do 5 and 5, or if you have sort of really antsy participants, you can even do 3 minutes, 3 minutes, 3 minutes. We do a bunch of those. I'm working on a kid study at the moment. We have a 3-minute blocks, but we do 5 of them, 5 times 3, in order to get that power sort of worked up.
Just on your point, I wasn't sure whether you were asking in that you don't acquire the images at the same time, but in the same session. So, you know, within minutes of each other.
Yeah, I was just confused, or I was a little bit worried about how you take, you know, when you take 2 photos of what should be the same thing,
Yeah.
but if you try to smush them together, surely they don't smush properly. I mean, do you have to warp things a lot to do that, or and just warping change anything, or is that a problem at all?
Yes, is the short answer. So there's a lot of very intelligent people working on the question of how to best warp one brain on top of another, because you can't just sort of resize and shift it around. Of course, you need to sort of change the size of structures and these sorts of things. When you do that, you preserve the signal strength. That's not a problem. But then, when you're overlaying my brain and your brain on top of each other, and we've warped them to each other, do we get a correspondence of regions? And so some of the ways we get around that is by smoothing our data. So we work voxel by voxel, but then we actually kind of smooth across those voxels a little bit. We smear the data in order to account for some of the inter-individual variability. And then just sort of accept that it's going to reduce our power, but it's not going to cause false positives. There are other ways around it if you're interested in a particular region of the brain, and then you would sort of say warp to that particular region, and you could do sort of gyrus by gyrus or sulcus by sulcus type of registration. But in terms of a whole brain study, it is a confound, but it's not going to invalidate the research.
And if you were going to conduct your own research, would you, in imaging, would you usually get someone to register for you, or you learn that kind of stuff? And there's lots of different registration techniques, and you just choose.
There's standard pipelines.
Oh, okay.
So there are a couple of big statistical packages. SPM, statistical parametric mapping is sort of one, and FSL, I don't even know what it stands for, but those are the 2 sort of main packages, freely available to the academic community, and they have sort of built-in algorithms for each of these steps, because there's actually quite a number of steps that you need to go through in order to get to those block maps.
It's a good point. I mean, even the simplest, what appears to be the simplest anatomical image, if you acquire your brain on one scanner and your brain on another scanner today, we would still have a bit of trouble matching those 2, because it's not a photograph of your brain. It's based on the water molecules moving around and pulses going in, and different scanners might do that slightly differently. Yeah. And then it might look slightly different even though you scanned it on the same day.
Yeah, that's right, because I'm part of the ABLE study, and they're also with ADNI, which is this international thing, and they try to match imaging things together.
In terms of multi-site.
Do that, yeah. But anyway.
I mean, people do it, and you can do it, but it's just important to remember it's not a photograph. There's issues.
It's machine specific, if you will.